This afternoon, I'd like to talk about some work that we did to put together a solver for differential equation systems, for dynamical systems, that makes use of both the deterministic solution for the situation when you've got a lot of particles in your system, for example, molecules in the traditional sense, or in my case, healthy cells, infected cells, and viruses. And coupling that with the situation when you don't have many of those types of entities in your system, and you need to model on a stochastic basis, where the random effect, the random nature of what event happens next, and then the random time delay until the next event become important. So we run into this situation a lot in some of the modeling that we do when we're trying to treat a disease and we're trying to knock out a bacterium or a virus, for example, and we treat long enough that the numbers of viral particles gets to be very small, and the stochastic effects could then become important trying to determine how long it takes to actually cure a patient, for example. So that's the motivation behind this work, and what we wanted to do was put together something that we could use pretty much off the shelf in our work, much like one would use ND-solve off the shelf and just expect all the pieces to fall in place. So that's what we did. So I will talk a little bit about what we put together and then show two examples. The first one is going to be a completely stochastic simulation of a gene regulatory network, because that shows the stochastic type of modeling that a lot of people aren't familiar with. And then the second example is flu infection, and that one does make use of both the deterministic solution using ND-solve and then the stochastic solution with a Gillespie algorithm, which is the part of the system that I wrote that works along with ND-solve. Okay. So that's the overview. Why do we have to do this? Well, the stochastics is important because biology is basically noisy. We've got variability between patients. We've got variability within patients. We have varying concentrations of metabolites in the body that cause different effects at different times. And we can't possibly model all of that exactly if we knew everything, and we don't know everything, so we make approximations, and using the stochastic methods help us do that. The first kind of noise about interpatient variability leads to Monte Carlo type simulations where you choose your parameters on a random basis around central values with a given distribution. I'm not going to address that problem today. The intra-patient variability, though, where you have to worry about things happening because of the random nature of molecular events, is what I'm going to talk about, and we handle that with the Gillespie algorithm. To put things in a little more context, traditionally in chemistry, we study reactions in a beaker where we've got billions and billions of molecules, and we can treat them as an ensemble as a time average with the differential equations and do that deterministically and get exactly the same solution every time we solve the problem. On the other hand, when we deal with reactions in a cell, or for example, viral particles in a patient, we have much smaller numbers. Instead of 10 to the 23rd, we're dealing with 10 to the 10th, 10 to the 8th or less. And there we use the chemical mastery equation, or the Gillespie algorithm, as our method of solution. So what we do then, to do this sort of modeling, it's a Poisson process, and we essentially throw the dice to find out what reaction is going to happen next, what biological event is going to happen next, and then we throw the dice again a second time to find out how long to wait. The probability distribution that we use for choosing the event is much like a roulette wheel type process, and I'll go into some details about that. And the probability distribution that we use for choosing the time is an exponential distribution. If you saw Roger's talk yesterday, a lot of the stuff that we've put together is going to be out in the next version as part of the random process functions. And I'm really looking forward to using them that way because it's going to be well integrated and it's probably going to cover a lot of cases that we didn't anticipate. Okay, we can skip that. So here's the first example. It's an autoregulatory gene network. What we have is your strand of DNA that gets translated to RNA. The RNA produces a protein, and the protein will dimerize. And the protein dimer acts through a negative feedback mechanism and binds with the DNA and keeps it from being processed to form more RNA. So we have this negative feedback loop. And the way we approach a problem like this is we write a set of reactions in Mathematica that describe each of these steps. We've got the transcription, translation, dimerization, the repression binding step, and we've also got degradation of RNA and degradation of the protein. And we can write the model this way, and this is a good visual check to see what's happening. And if you've had any experience with System Modeler yet, and you look at the models graphically, they look very much like this type of structure as well, where you've got entities and you've got the reactions gluing the entities together. Okay, so let me load the package that we use that lets me write the reactions and then parse them and turn them into differential equations and everything. I'll put that away for now, we don't need it. Okay, so here I've written the reactions as Mathematica expressions, reactants on the left, an arrow with the rate constant, and the products on the right. And I've also defined my parameters for the model here. And then I'll call kinetic model, which will give me the variables of the system, the reactions, which are basically these, but it undoes equilibria as two separate fundamental reactions. It gives me the stoichiometric matrix, it gives me the rates for these reactions, and it gives me the set of differential equations. All of these are needed to do the deterministic modeling or the stochastic modeling. So I get everything from one authoritative source for the model, and I'm able to go either way or both without having to worry about forgetting something in between or losing it. Okay, so here I'm writing out the differential equations that were generated. I have to write my own initial conditions because those are always problem specific, and they often vary a lot from experiment to experiment, whereas the model itself is constant. So I'll define my starting conditions. The starting conditions are 10 gene molecules, and everything else is at 0, not present. Okay, so let's check the overall behavior of the system. If we do the deterministic solution, we can see that we start out with just 10 genes, and it produces protein here, and the protein dimerizes and forms a lot of dimer. The other species in the system, the gene and the RNA, are all very small. And if we look at the first 30 seconds of the simulation, here's the protein dimer, here's the gene coming down. But things like the protein, that's a very small amount. The gene itself, on time average, ends up being less than 1 molecule. And that biologically doesn't make a lot of sense. How can you have less than 1 molecule? And this is an artifact of the type of solution here. So this is one reason, another reason why we want to do a stochastic solution. Okay. So if we look at it, yeah, here's the details, looking at it, how the gene comes down to less than 1 gene on average, and the RNA is also way less than 1 RNA molecule. Okay, the information we need to do the stochastic simulation is shown here in this table. We've got the individual reactions that we get to choose one from by the throw of the dice. The stoichiometric matrix, this tells me how many molecules are being changed from one side of the reaction to the other in that step. And then this is what's called the propensity. It's essentially the concentration of reacting molecules and the rate constants that's going to tell me how fast this process is going to take place, and tells me how long I have to wait until the next event. Okay, so to get those propensities, we need a function to calculate that. And so we call this function here, define propensity function, that will let me define everything. This is the usage statement for it. It takes the variables, the rates, my time variable, my parameters, and the function that I want to use to call it propensity, a name for it. So I'll call the function. I'm going to define this will define the function propensity for me. And one of the things that it does is it gives you the usage statement for the function it just defined, so you know how to use it. And here we're going to say at time 100, I'm going to define a state where I've got 2, 10, 8, 1, and 79 molecules of the different species in my system. That's essentially my state vector. And then I'm going to calculate the propensity for that state and at that time. And here it is. So this tells me what the probabilities are, basically, the relative probabilities of each of the events that I can choose from. So what we do then is we generate an empirical distribution from those propensities. And we can take a look at that, and it looks like this. The first reaction here, which takes the gene and produces RNA, very, very low likelihood of happening, whereas something over here, the gene binding with a protein dimer to do the inhibition, is the most likely thing to happen. And what we want to do in a probabilistic sense is look at the cumulative distributions here to find out what we do next. So it's basically drawing a random number between 0 and 1, coming over here to see where it hits the cumulative distribution and looking down to find out what event we're going to use. So we do that here, and in this case it drew that last step, and it changed the state change. It would be from this state vector to this one. We lose a gene and we gain a bound gene, and we also get the other product here. I don't remember which one that is. You know, if we do it again, we get a different reaction and a different state vector change. Then what we do is we use the exponential distribution to get the probability function that we're going to use to draw the time step, and we can draw a time step. And here's the exponential distribution, it's cumulant, and drawing the time step here says that I need to wait 0.02 of a second until I repeat the process, and so the time then increments from 100 to 100.002. So that's an individual step in the stochastic process. Okay, so to do this, then what I have to do, first of all, is export my model and all the definitions to the solver that runs in batch mode, because some of these simulations can take many minutes to several hours depending on how big of a system and how many repetitions we want to run. So I've written out the files in Mathematica format, and I'm going to open the notebook now and basically run it so we can see what happens. These steps here load those files I just read, so define the model and the parameters, and I specify the name for the results. I give some information here, the user would, about the particular scenario. We're going to start at t equals 0. We're going to go 5,000 seconds. These two parameters determine when I switch between deterministic and stochastic. I'm going to summarize my state every 5 seconds and write it to the output file. And I'm not going to restart the situation. I put in the ability to restart in case you need to debug something or you want to be able to reproduce results and maybe run it longer. Okay, so we basically just say run the package, and it's done. This is a very fast simulation. So we'll come back here, and I've just done that, so I don't need to do it that way. Okay, so let's read the results in, and there's just one result because I did just one simulation. The results that I get back are actually very much like the output from ND-solve and can be used exactly the same way. It's a piecewise composition of interpolating functions, so you can just use that like anything else in Mathematica. You can plot it, you can integrate it, you can differentiate it, use it to calculate concentrations of things at specific times. Okay, it's very big, but it's something that's quite usable. I also returned the specific parameters that were used for that particular simulation, so you know exactly what you had when you started. So we'll pull that out and look at the bug. I apologize for that. Okay, Let's see if I can pull up the notebook and show you what it looked like when I put it together. Okay. Not quite as nice as the current set of slides, but I think it will make the point. Okay, so here are the results of the stochastic simulation. This is the gene protein dimer complex, protein dimer, and so on. And you can see that the gene comes down. It never, sometimes it goes to 0, but it's 1, but it's always, a reasonable biological value. It's not a fraction of a gene. And we can see that it actually hovers right around the deterministic solution. But this one here is much more realistic in terms of what's actually happening. And here it is for all the different species. I almost hesitate to go back and run the slides, but we will give that a try anyway. Okay. Okay. The next example I want to show is flu infection in an individual. And here what we have is we have healthy cells and virus that the virus infects the healthy cell and forms an infected cell. The infected cells produce more virus, and the infected cells die and the virus dies. So that's the basic model, and here's the graph for it. Here's the set of reactions that we write, define those, define the differential equations and everything. And this is really, really embarrassing. I'm sorry. We'll go back here and see if I can at least walk you through this. Okay. If you solve the system deterministically with the differential equations, we have the virus starting out here with no infected cells, and all the cells are healthy, and we start infecting cells, so the infected cells come up and the healthy cells go down. This is a target limited model, which means that the healthy cells don't reproduce. Okay, so they're not replaced, and they will decay away, and when the number of healthy cells gets small enough, there are not enough around to get infected, and then the infection starts to die off because the infected cells do die and are not replaced. The model fits the experimental data pretty well. And if we do a long-term simulation out here, we can see that deterministically, the concentration keeps getting smaller and smaller. The amount of virus gets smaller and smaller, but it never goes to 0. And if we calculate what the concentration would be for just one virus in one cell, that's way up here at this level here. And the deterministic solution just keeps going on its merry way, subdividing and subdividing infinitesimally small viruses and cells, which is not realistic. So again, another reason why we want to do the stochastic simulation. Here's the information for the stochastic simulation for this model. And what we have to do here that's different is determine when we're going to switch over between deterministic and stochastic. So the way we do that is do a simulation that's going to be pretty much entirely stochastic for a short period of time to find out what the appropriate transition is. And that we do by choosing when the variation in the observables of interest to us, the variables of interest, are varying about 1%. And here I did 10 simulations, or 25 simulations, and I wanted to find out when the variability across those simulations was about 1%. And when we look at it, it occurs out here at around 0.75. We can do the calculation to find that out exactly, which I think is on the next slide. It occurs at about 7.5, and so that at t equals 7.5, I then compute the propensity, which is around 30,000. So that's what I set that threshold to. okay, I would run the simulation just like before. This one takes about half a minute to run, and I am not going to risk another embarrassment. So here are the results that we get. We read the results in, and I'm just now plotting the tail end of the simulation. And you can see that it starts out, they're all the same up to about this point here, and then the random nature takes over in the process, and we get lots of different different evolutions of the process, resulting in different cure times. And this is what we're interested in, because we would want to know, in a medical setting, how long do we have to treat to reach cure? And how do we account for the variability within patients and between patients? So we would take these results, and we could calculate a number of different distributions to find out which ones have the appropriate shape. In this particular example here is a histogram of the actual data of the times to cure, and I computed a normal distribution, a log normal, and a smooth kernel distribution to see which ones seem to fit the data the best. They're all fairly similar in this case. It is important to note, though, that the distributions generally are not symmetric, and they often have a longer tail to longer times, that you see a fair number of patients that take a long time to cure, rather than and a much smaller number of those that cure quickly. But we can use these distributions that we fit to the data to get the quantiles, for example, of how long it takes. And this calculation here using quantiles says that if I want to find out how long it takes to cure 95% of the patients, it'll take about 10.5 days to do that, or for the virus to die out in this case. And to do 99%, it takes a little over 11 days for 99% of the patients to clear the virus in this example. Okay, implementation details. Originally this was all done in Mathematica, but the stochastic simulations for some of the problems we were working on would take 12, 18 hours to do, and that's too long to do and try to understand your system and make changes, and also just simulate thousands of patients when you're trying to simulate a clinical trial. So we took the stochastic part and rewrote it as C code, and we isolated only the time-dependent part of the model is a separate piece that Mathematica would then write in C at runtime, and then the C program shell would incorporate that. It would get compiled and then run to do the simulation. The parameters for your particular simulation are passed through a data file, so that's very easy, doesn't require any changes to the code. So we basically minimized the problem-specific time-dependent part of the code and incorporate that at runtime to do the simulation. So that gives us then a very fast way to do these simulations and bring the results back into Mathematica and carry on as though we did everything in Mathematica from the very beginning. And that's pretty much it. Improvements that we could make. Right now, this system assumes that everything is operating on the same time scale, but that's not always the case. Sometimes you have some reactions that happen very, very fast and very often, and others that are important but happen much more slowly. And so we need a different algorithm to handle that. For example, the tau leaping algorithm that lets the very fast-acting reactions happen sort of on an average time scale, an average effect, and then we model the slow things more stochastically.